Welcome everyone. So today we're attending this webinar to talk about our new automated nanoparticle system and how it can be used for formulation library generation for vaccine development. So let's get started. So some of the things that I want to cover today are basically let you know what we do here at Dolomite. So just a brief introduction to the team. Why do we need vaccines? An overview of the process of vaccine development, introduction to microfluidic technology and lipid nanoparticle production, and finally introduce you to the ANP system and how that works with a brief demo, followed by again, a little more discussion around formulation development and selection. And finally, we'll conclude and take questions in the end. So please reserve your questions for the end of the webinar. Thank you. So here at Dolomite, we are a team of chemists and engineers who have a focus to produce high-performance microfluidic platforms which allow precision control over size, shape, and structure of your final product. We focus on a broad range of applications spanning over various different industries, not limiting to, but including biomedical and pharmaceutical industries. We supply standard products such as particles, specialist chemicals, and emulsion stabilizers. And if you do like to know more, please do visit our website. We advise customers on microfluidic setups and processes. So if you're new to microfluidics and you want to implement this technology in your current lab process, we are happy to help in that respect. And if required, we can undertake proof of principle projects for customers ahead of any equipment purchase. So why do we need vaccines? We know that the economic burden of disease is a serious global issue because just looking at the last 20 years or so, we have been hit with various pandemics and epidemics. We're currently undergoing one with COVID-19. And over the last century, vaccines have really helped eradicate many of the life-threatening diseases. And so this is just some of the ones we've encountered in the last century or so. And of course, it's not just limited to these. And we can see with the green cursor, when a vaccine has been introduced, the disease has been eradicated and limited. So they play a very important role in how these diseases shape our world. The lifespan of diseases is directly linked to the introduction of the vaccine. COVID-19 has by far broken the record for vaccine development when it was first introduced into the world, in the environment, at the back end of 2019, 2020. In the same year, we seem to have developed a vaccine, and we can see that is the quickest we've ever been able to introduce a vaccine onto the market. So vaccines really help shape healthcare worldwide, and continued research and development is key to maintaining this. With the current picture, we have COVID-19, which has affected all of us in one way or another. And we have currently around 91 vaccines which are in clinical development. And many of these have already been rolled out worldwide. Some of you may have already gotten your first or second doses. I'm still waiting for mine. And a further 184 are still in preclinical development. So there's a big effort driving towards these vaccine developments. And recent news coverage has also said they want to develop further vaccines for new emerging variants of this virus to further protect everyone. So there are various different types of vaccines that are available. You have your virus. You can have it live attenuated, inactivated. You can use the viral components, so protein subunits, or you can use the genetic material, the genetic code that the virus actually holds. So here is just, a very brief list of some of the key leading pharmaceutical industries which have paved the path for vaccine development. And for example, in the UK, we have the AstraZeneca vaccine, which is quite popular, and the Pfizer, and also the Moderna vaccine as well. Whereas in the EU, some of the other ones may be more popular. So whilst there is a lot of ongoing research to identify key vaccine targets, the way to package and deliver them also needs consideration. So let's look at how the process of vaccine development actually happens. So this is, again, a very brief overview, but usually you would identify a target pathogen. In this case, it could be a virus. You would sequence them. So very early on, in January 2020, the genome was already sequenced and it was shared worldwide. I think something like 6 different sequences were shared, and it's continually being monitored. Once you have that information, bioinformaticians carry out some of the selection of vaccine targets. So once those are identified, these are then further cloned and expressed and purified to basically help do further clinical trials and rollout. However, before any of that happens, there is a step which is called formulation, and this is where the packaging of your vaccine happens. So this is to protect it and be able to deliver it to the human or any of the animal testing that may be taking place. So it's a key process in vaccine development and often overlooked. And a lot of the research is focused on the first 4 steps, and the 5th, for example, there is still a need for further development. So this is where we are bringing in our ANP system to help with this step. So let's look at some of the current challenges for formulation development. At the moment, there are no advanced formulation screening platforms. There are no off-the-shelf ready-made solutions that you can simply package your genetic material or vaccine targets in. There are no R&D scale robust platforms for our lipid nanoparticle production. There are no controllable technologies for developing formulations, and there's certainly no automation in any of the existing platforms. Some of these existing platforms are listed over here, and some may be familiar to you and maybe using these in your experience with in the past. And certainly most of these methods potentiate they are laborious. They have huge quantities that you have to work with. We're talking milliliters to liters. So there's a lot of wastage that comes out of that. They are unreliable. They offer poor encapsulation, and there's hardly any control on how you can manipulate some of the parameters, and they have poor reproducibility because of their batch-to-batch process. So there are various different challenges that we face with current and existing platforms for lipid nanoparticle productions. So with microfluidic technology, some of you may be aware of what microfluidic technology is, but I'll like to explain a little bit on what that is. Lipid nanoparticles can be produced using this technology by running 2 fluids in a microfluidic chip. So these lipid nanoparticles are formed using a process called nucleation and growth. Lipids can be dissolved in a solvent of your choice, so this could be ethanol in this case, or it could be chloroform, methanol, or a mixture of different solvents. And these lipids are hydrodynamically flow focused in a chip by an antisolvent. So the antisolvent is usually an aqueous buffer, for example, and contains your API or your genetic material. If your API is hydrophobic and can be dissolved in the lipid phase, you may be able to introduce that in the organic phase. So you have lipids in ethanol, and they have your aqueous phase, and these are hydrodynamically flow focused in the chip, which causes the mixing of the 2 fluids, allowing for the liposomes to assemble. So the particle size is controlled by various different parameters, but one of the things which is key is flow rate ratio and the total flow rate. What I mean by the flow rate ratio is essentially the flow rate between the organic phase and the water phase. So the easiest way I can show this, if you look at the bottom 2 images on the screen, we've got the first one where we've got the organic phase and the aqueous phase, and we can see a thin sort of a line forming in the collection channel. And with this one, we can say it has a high flow rate ratio, which means, for example, the organic phase may be 1 ml per minute and your aqueous phase may be 10 ml per minute. So it has a high flow rate ratio. Whereas the second one, it's a lower flow rate ratio. So it could be, for example, a 1-to-1 ratio where they're both flowing at 1 ml per minute. So simply by manipulating the flow rate ratio, you're able to control the resulting lipid nanoparticle size. So this is greatly important for users to be able to control that, especially in real time. And you're also able to control your total flow rate. So if you want to have higher throughput, by maintaining your flow rate ratio, you can have both fluids running at a much higher flow rate. So that also is an important aspect that should be able that the user would like to control. So the API can be brought in via the continuous phase if it's hydrophilic, or the inner phase, which if it's hydrophobic, or it can also be encapsulated afterwards via active loading methods. So it's completely up to the user. A few of the key advantages of using microfluidic technology is that it offers high encapsulation efficiency. It has excellent PDI because it's highly reproducible. So the self-assembly of these liposomes happens more or less exactly at the same time in the same environment. They undergo the same process. And it also allows the user to use low sample volume. So this is greatly important for precious cargo, or anything that you're working with is highly valuable. And obviously you want a technology like this to be scalable, which it is. So again, you're able to work from an R&D scale to a much higher throughput. So just to share with you some of our system data, so there is an application note that we'll be releasing in the next month or so. So one of the things we've done here is altered the flow rate ratio between the organic and the aqueous phase, and simply by doing that we can see that we've been able to manipulate the lipid nanoparticle size anywhere from 20 microliters to 150 microliters. And this is not just limited within this region, we can push beyond this. So again, it depends on the lipids that you use, the solvent that you use, entirely dependent on your formulation as well. And we can see that with the PDI, it's less than 0.2, which is excellent. And the corresponding image on the right-hand side is the DLS data, the Melvin Master Sizer, which shows these nice peaks, again, shifting the size of the lipid nanoparticles based on the flow rate ratio change. So what is our automated nanoparticle system? It's the first of our new range of systems that allows the user to develop protocols and produce lipid nanoparticles. It allows you to have that flow rate ratio control, which is very important. So again, it's user defined, it's up to the user. It allows you to have control over your total flow rate. So it can go up to 15 ml per minute in protocol mode. Once you have optimized your parameters and you want to have much higher throughput, you can simply turn it into a more of a continuous mode and go through bulk production, and it can go beyond the 15 mils per minute. If you have precious cargo, for example, it can work with volumes as low as 200 microliters and collect samples as low as 200 microliters as well. It allows you to have further control over your final sample by allowing you to have a head and a tail cut. So as your sample is mixing and is traveling to the collection vial, you have the option to chop off a small amount at the front and the back, so you're collecting a very nice hard cut, which has no contamination or dispersion that may happen during inline travel. Between each run, there is no risk of cross-contamination because after every experiment, there is a wash cycle. So everything is washed before the next experiment commences. Each experiment is around 3 minutes long, and again, this is actually dependent on the user as well, how quickly you load your sample. That's the only manual bit that you may have to do. So on the top right-hand side, we can see a picture of the whole system setup. We have the pressurized input store. So this is where you would put your driving fluid, which drives your sample towards the chip. You have your automated sample valve. This is the valve that allows a release of your sample for multiple experiments. You have your your Metos pod pumps, which are actually the syringes which is pushing your fluid towards the microfluidic chips, and you have your automated collector as well. With microfluidic chips, you have a range of different options available. All of these essentially are designed to create nanoparticles. So they have pros and cons for various different parameters, but essentially you can use any of these chips that are available to you to produce lipid nanoparticles. So how does the system actually operate? So your nanoparticle precursors, which essentially is your lipids in solvent and your cargo in buffer, I refer to them as nanoparticle precursors, are loaded in separate sample loops. So we have the ASV, which is your automated sample valve, and we have these sample loops which are cased here. So this is where your sample would be retained. So they can come in volumes of 1 ml, 5 ml, 10 ml, or it can even be customized. And here you would manually inject an enough nanoparticle precursor for several experiments can be loaded in one go. So you can see me injecting some blue dye, for example. So this blue dye is going through that fitting and into the sample valve. So this is how you would load a sample. So if you're doing multiple experiments, you would load the total volume for those experiments. So the high performance pumps feed nanoparticle precursors from the loops through the microfluidic chips into the collection unit. The system is designed to ensure all nanoparticle precursors meet at the correct time. So your ASV is releasing your sample from sample loop 1 and 2 at the correct timing. So when it meets at the chip, it's able to do the hydrodynamic flow focusing and the mixing to allow assembly of your nanoparticles. And the collection valve also allows you to have the accurately captured hot cut, which may be important to some. So the samples are collected in a series of vials without manual intervention, and in between repeats, the fluidic pathway is washed before commencing to the next collection. So again, a little graphic to show an overview of the whole process. So we have the Metos quad pumps. These basically draw fluid from your driving fluid, which sit in the pressurized input store. There may be just some buffer, which corresponds to the cargo buffer as well, or it could be the solvent that you choose for your lipids. So it's completely up to you. This is simply a driving fluid which pushes your sample into the chips. So once the syringe is filled with that, these syringes push the sample that is loaded into the loop. So here's your loop, which has your sample in there. These then drive your sample into the microfluidic chip. So this is your first microfluidic chip where the two different fluids will mix. So your lipids in solvent and your cargo in PBS. These then have the option to have a further dilution step. This is again optional. The user may choose to bypass this completely. That's absolutely fine. But this just allows flexibility if you wanted to introduce that dilution to allow further growth of your nanoparticles. And then you have your collection valve, which switches between waste and collection to enable that hot cut of your sample. So if we look at closely with the ASV has various different functions. It has obviously the loop. It has the manual injection where you fill your sample, which puts it into this loop, and it goes into to and from the chip and the waste. So when you're injecting your nanoparticle precursor, it is in this position. So as you inject, it sits in this loop, so it fills this loop. And once it's ready to dispense your nanoparticle precursor towards the chip, it will be in this configuration. So it will disconnect and from the manual injection, and you would drive from the pump into the chip. So there is a new version of FCC with protocol editor that accompanies the system as well. There are fluidic pathway. The fluidic pathway generally is fixed for up to 3 inputs, but is flexible for different chips. The user can easily set up a list of experiments varying the flow rate ratio, the total flow rate, and the sample size. And the system can automatically calculate all reagent volumes for you. So when we are interested in working in continuous mode and we want to come out of protocol mode, and this is usually when you have optimized all your parameters and you're happy with the size that you're getting for a given total flow rate and a flow rate ratio and so forth, you can simply switch the system into continuous mode and have your bulk production. You can get all your results exported because of these log files, and you can keep track of that way. This also saves your protocols. So if you have multiple users, they can have their separate protocols available and they're ready to go when you next use them. You can import experimental tables that you set up offline. So this is just a simple Excel CSV file that you can import and export or copy, and you can create your experiments offline. So you don't necessarily need to have access to the FCC for that. So when you open up the experimental table on your FCC, so if you edit this, a pop-up like this will come up. So it allows you to set your flow rate for your nanoparticle precursor 1, your nanoparticle precursor 2, and if you are using the dilution. So there is an option to use the dilution pump. So you can select yes or no with the drop-down menu there. And based on the flow rates that you set between these three, it determines the flow rate ratio for them. So you might have a 1-to-1 ratio. So for example, here it's 1-to-1-to-1 when it's 1,000 and 1,000 and 1,000. And how much sample would you like to process during an experiment is dictated by this column here. So you decide what volume you want to use for nanoparticle precursor 1, for example. And based on the flow rate ratio on your flow rates, the system can automatically calculate the volume that you would require for your nanoparticle precursor 2 that you would need to load in your sample loop 2. The system will have, once you've started your experiment, it will have a pop-up window which tells you how much you need to fill each of those loops. So it has a lot of automation in terms of calculating things for you. And then, like I mentioned before, you have the option of a head and a tail cut. This simply means if you have a 2 ml fluid packet, so if your total sample volume is going to be 2 ml, you're only interested in the hard cut, you can remove the front or the back 500 microliters or any volume that you require, or you can do multiple different tests to check which part of that sample you would like, or you can completely choose to ignore this step and simply enter 0 and move on. So now I'm going to move on to just sharing with you a little system video talking through how the system actually works, and I've just used some few dyes for demo purposes. So just bear with me while I switch to that. So I'm going to show you how the system works. We've got in front of us 2 Mitas quad pumps, pressurized input store, the automated sample valve with the sample loops attached. So in this case, we've got 5 ml, and our automated collector. We've got the 2 microfluidic chips. So the first one which will do the hydrodynamic flow focusing, and the second one which can introduce an optional dilution step. For today's demonstration purposes, I'm using 2 dyes. These are just basic food dyes. We've got the blue one and the yellow one. So once we mix them together, we should get a nice green sample collected at the end. So if I take you now to the flow control center, which accompanies the system, all of the components are connected and should show up in the devices tab. So various different windows are open, most of 3 of which belong to the 3 syringes, the Gilson collector, and the ASV. When we want to run experiments, we usually go into the protocol mode and we go into the experimental table. So I've already pre-filled this table for us today. I'm going to run 4 different experiments at a total flow rate of 3 mL per minute. I've chosen not to use the dilution step, so this remains 0. Loading capacity for one experiment is 500 microliters. Because I'm running 4 different experiments, I'll be loading a total volume of 2 mL in each loop, because the flow rate ratio is also 1-to-1. So again, flow rate ratio is determined by the input values in this experimental table. I've decided that I wanted to do a head and tail cut, so I've decided that I want to do a 250 microliter head cut and a 250 microliter tail cut, which means the total fluid packet of 1 ml would remove the front 250 microliters and the back 250 microliters, so I should
collect a 500 microliter sample in my automated collector. So let's go ahead and start the experiment. So I want to run these experiments and I click on start. So once this happens, the output script here tells you exactly what's going on. So at the moment it's telling me that it's priming the system. So that just means the syringes may be filling or the fluidic line may be washed. So yeah, so at the moment it's telling me the pumps are being filled. This usually happens, so if I take you here, it happens with the driving fluid that's located in the pressurized input store. So in this case, I'm using buffer, just 1 times PBS buffer, to fill all 3 syringes. So this is basically going to act like a driving fluid. Now the system is prompting me to load my nanoparticle precursors, in this case the food dyes. So it's telling me, please inject 2 ml into sample loop 1 and 2 ml into sample loop 2. This is because we're loading the total volume of all 4 experiments together. And because it's a 1-to-1 ratio, 2 ml in the first one and 2 ml in the second one. So I'm going to go ahead and do that. So the first one is nice and yellow. Hopefully the camera will be able to display some dye going through the tubing. So I pre-filled these syringes. So now the first one is done, and it's gone into these sample loops. And hopefully you can see the blue one is filling the second sample loop. Okay. So once that's done, you simply press OK on the pop-up dialog, and we just heard the automated collector move into position 1 to start collecting the first sample. Because we're doing 4 different experiments, I have lined up 4 different vials. So these are just simple 1 ml cryo vials that we use. So you can probably already see the dyes are traveling towards the chip where they'll mix, and the final output is green. So we've got the blue and the yellow going towards our chip. And because we've told the system to remove the front 250 and the back 250, it's taken some to the waste, and now it's starting to collect. So we expect about 500 microliters to be collected. So that's the first experiment done. Now the system is undergoing a washing cycle to remove any contamination of the fluidic line in preparation for the second. So maybe you can see this in the camera, maybe you can't, but we've got 500 microliters collected in this vial. So that's the first experiment done. And now we're waiting for the lines to be washed. So you can see where we had fluid that was colored in the fluidic line. That's all been removed. Some may be wondering, what does that mean? What happens to the dye that was loaded for all 4 experiments? The wash cycle usually washes everything else without disturbing the sample loops at this point. So they remain intact, so the next one will begin shortly, and that will release the next set. So again, the Gilson, the automated collector, has moved to the second position to start collecting the second experiment, and the dye will eventually start releasing and go through the chip again. So that was a pretty short cycle for the first experiment. It took us a couple of minutes, in fact even less than 3 minutes. And some of that depends on how quickly you sometimes load your sample into the ASV as well. So again, the dyes entered the first microfluidic chip. It's gone through. We can already see it turning green, and very quick, yeah, so it's going to start collecting now, which it is. Hopefully you can see that. So the collection has now finished. That's now gone to the wash pot to wash the nozzle. And again, we've got a nice 500 microliter sample. So will carry on doing this until it's finished with all the 4 different experiments, and you are ready to analyze them downstream using a DLS or whatever UV vis, for example, that you may be using. So it's literally as simple as that. And once all of your experiments have finished, the whole system will be washed, including the sample loops, and it will remain ready for your next set of protocols, for example.
Yep. Yeah, so again, just taking you back to the system data again. So this is the application that will be released shortly in the next month, and it was produced using that very system that you saw on the demo. And by using a very simple formulation with 1 cationic lipid and 1 neutral lipid mixed in ethanol, and we simply crush that out with some PBS buffer. So by simply manipulating the organic and the aqueous phase flow rate ratio, we were able to get a nice control over the lipid nanoparticle size. So just talking a little bit more about formulation development and selection, going back to RNA vaccines, they are a promising therapeutic tool in the fields of vaccine development and protein replacement therapy. Why that is? Because they are non-infectious, non-integrating platform. There's no potential risk of infection or insertional mutagenesis. Their half-life can also be regulated. However, they are degraded by normal cellular processes, which essentially leads us to wanting to protect them from being degraded, and therefore we need to find delivery systems. And although there are various different delivery systems available, and some of them are nicely portrayed here in this figure over on your right, lipid-based delivery systems and polymer-based delivery systems do take the lead in this kind of aspect. So if we focus on lipid nanoparticles in particular, they represent one of the most advanced platforms for messenger RNA vaccine delivery. And this is because they have several different advantages to be used as vehicles for vaccine delivery, because they are stable, they increase the stability via encapsulation, they are non-toxic, they are flexible, they are biocompatible because they resemble some of the cell membranes in the body, they're completely biodegradable, they are non-immunogenic for systemic or non-systemic administrations. They also increase the efficacy and the therapeutic index of your encapsulant. They reduce the overall toxicity of the encapsulated agent if it is toxic in the first place. They also reduce the exposure of sensitive tissues to any toxic API or genetic material for that matter. They have site avoidance effects, and they offer flexibility to couple with site-specific ligands to achieve targeting. So they can be coupled with specific proteins, antibodies, and things like that. But when you're designing your formulation, it's important to consider the route of administration as well. So where you want to deliver your final vaccine. This is important because the volume that's required in various different areas of the body varies. And if that varies, you also have a high reliance on encapsulation efficiency and on the actual dose that you're able to reliably deliver to the site. So that needs to be considered very carefully. And again, the size correlates in with all of the above things that I've just spoken about. So depending on the route of administration, the particle size is very important as well. So you could require anywhere from 10 to 600 nanometers, and this system is capable of achieving these size ranges very easily. We've already sold multiple systems to several customers, and we've had reports of 99% encapsulation efficiency of RNA with a really nice control over their final particle size with a very low PDI. So we're really excited to be working on this system. The other key things worth remembering when you are designing your vaccine formulations is 5 of these key components which keep coming up when you're designing a formulation. So cationic lipids are an obvious thing because they are positively charged and they form electrostatic interactions with the negatively charged material. So they work really well for encapsulating anything that's biological. In the recently, ionizable lipids have gained much more popularity because they can change charge at acidic pH, so they become neutral at physiological pH, and they interact with endosomal membranes during the fusion process and release the RNA into the cytosol. So they are gaining much more popularity in conjunction with cationic lipids. So it's important to consider which one you want to go ahead and use. Helper lipids offer distinct functional properties. They enhance the stability, promote cellular uptake and destabilization of the lipid bilayer, and thereby improve efficiency of your RNA delivery. Pegylated lipids are known to increase and extend the circulation time of your lipid nanoparticles due to their steric barrier effect. And cholesterol provides and contributes to stability, structural integrity, and physiogenicity of the lipid nanoparticles to membranes. So we've just listed a few of the most commonly known cationic lipids, ionizable lipids, helper lipids, and pegylated lipids. And depending on how you use this formulation, you may be forming any of these particles, lipid nanoparticles. You may end up forming a liposome or a lipoplex, where your RNA is sort of attached to the surface of your liposome, or a lipid nanoparticle where you've encapsulated your mRNA. So very important to know how you're going to be working with your formulation, but this platform technology sort of offers you that flexibility to optimize and develop your formulation libraries. So just to conclude what we've spoken about today, so lipid nanoparticles can be produced by our ANP system. It's not limited to just vaccine therapy, but you can also use it for cancer therapy and various other things as well. And it's also not just limited to producing lipid nanoparticles. If you so wanted, you could produce polymeric nanoparticles and other types of nanoparticles as well. So this is the only existing R&D scale platform which allows users to optimize and develop their formulation and produce libraries which they can do downstream analysis with. It pairs microfluidic technology with automation to create the next generation of library development platforms. And we're continuously innovating and developing new products inspired by our customers. So we would love to hear your thoughts and feedbacks or any questions that you may have. We would love to hear from you. And lastly, I've mentioned this already, we do have an application coming out shortly with this system. So again, keep an eye out for that. So thank you for listening. I will take any questions. And I will be answering them, I think, live, talking to you. And also, there are 2 handouts available, so please, feel free to download these for your reference. So just give me a minute while I look in the questions tab, and I'll get back to you. Okay, so I think there's a question here asking what is the maximum flow rate that can be used. Like I mentioned, when you're in the protocol mode and you're doing designing your experiment, it can go up to 15 mils per minute. But when you use it in continuous mode, you can push it beyond that, so it can work more than 15 mils per minute. So there's no maximum flow in that mode. Okay, so there's another question here. Can we use different NP precursors 1, keeping precursors 2 constant to create variations in nanoparticles? Again, yes, you can do that. You can vary that exactly as you said. It's completely flexible, and you can pick and choose what volume and how you want to differ the volume between NP precursor 1 and 2. So completely up to the user how you want to do that. And then we have, what are the working temperatures? Can you work with solutions at higher temperatures? So at the moment, this particular system does not have temperature control, but we are, because this is the first in the series of this library generation systems, we are going to be releasing where we implement temperature control in the future. Okay, so we have another one. What is the maximum total flow rate that can be reached? So in protocol mode, this is 15 mils per minute, and in continuous mode, there isn't really a limit, so you can push it to 30 mil per minute, for example. It's going to work in a continuous mode, so the syringes are going to have a handover process. And it's asking, is the pressure monitored during the process? Yes, the pressure is monitored during the process, and you can extract that information using the log files as well. For bulk production, what is the maximum volume that can be collected? So when you're doing bulk production, so this is the continuous production, you would bypass the loop. So where you have your pressurized input store holding your driving fluid, you would simply load your formulation there. So your NP precursors would be loaded directly into the pressurized input store. So there's no limit on how much you can process, and there's no limit how much you can actually collect at the other end. So it's just a matter of you changing bottles, for example, have a big collection vial, for example. And what about temperature? So like I said already, it's not implemented in this particular system, but it's something that we're looking at in the future. Any information about the price of the instrument? So it's quite a modular piece of equipment. And it's quite customizable. So I would suggest to get in touch in order to get a little bit more information on the pricing. Okay, thank you very much. Can we buy only the microfluidic chip? How much is one chip only? So yes, you can buy the individual chips if you would like. I'm not too sure about the pricing because we're in different countries and everything like that. So I would again ask you to get in touch with that regarding that. Are there any droplets involved in the microfluidic device? So we offer other systems where you can make droplets. With this particular system, our focus is on creating nanoparticles by mixing off two fluids. So in this particular system, at the moment, we've designed it to produce nanoparticles, but we envision that future systems can be customized to produce droplets in this way as well. So it can actually potentially be used to produce droplets as well as producing nanoparticles. So how the organic solvent will be removed. So once you've collected your final sample, I would suggest you do some dialysis to remove the organic solvent, so you can get some membranes with different cutoffs. And that should, within a few hours, you should have your solvent that you can remove very easily as well. So why are you using higher amounts of organic compared to aqueous phase? Again, this is just dependent on the formulation. We use a very, very basic 2-lipid formulation, and wherever we could get control. So a nice thing to use is some design of experiments, which allows you to design your experiments in a way to find where the trends are. And depending, again, on the concentration and the flow rate ratios and all of that, it will give you a place where you can find the trends. And we found that by changing the organic phase in comparison to the aqueous phase gave us a nice control over the final lipid nanoparticle size. And then we have what is the minimum and maximum particle size that we can make. So already you could see that I had 20 nanometers. We can go as low as 10, I believe, and we can go as big as, I don't know, a couple of hundred nanometers. All depends on various different parameters. So we're talking about the concentration of your lipid. If the concentration is high or low, whether you're going to dilute your sample in line or not, all of these aspects contribute to the final particle size. So we can make them as large as possible. So we can talk, let's say, 800 nanometers, or as small as possible as well. So we have What is the biggest difference between Dolomite and Precision Nanosystems? That's a good question, and I think our system is unique in the way that we offer this R&D scale platform for you to produce library of your formulations, so you can have lots of different samples with a push of a button, and you can walk away from the system and you can come back 30 minutes later and you'll have your samples ready to carry out downstream processes. And obviously there's no sort of licensing or any of that aspect to the system as well. So it's completely user-friendly, flexible to your needs, very customizable and easily built upon. Has the DLS detected some larger particles in intensity that were not detected in number? That's a very good question, and it's something that we always investigate as well. So we usually look at number, volume, and intensity, and our aim is to always look at peaks where you have double peaks or a single peak. Again, depending on what you're looking for, we usually focus on reporting results which show a single peak in all three measurements. So if you have double peaks, then you perhaps need to consider if your population's got different sizes there. But any of the data that will be reported in the app notes will usually be reflective of the intensity and the number having a single peak. So how about microspheres? So with this system, like I said earlier as well, you can potentially make droplets as well, but we have aimed this to be in the nanoscale, but there's nothing stopping anyone from using it to produce droplets or microspheres or microparticles for that matter. So, and we do have some other dedicated systems for microspheres and microparticles. What is the difference between DLS and Zeta Sizer, which characterization? So essentially, it's just the way we name the DLS, which is the Malvern master sizer. So it's basically the same thing. The idea is to have dynamic light scattering analysis, which allows measurement of your nanoparticles. Usually we would recommend the use of using a Zeta Sizer or a Malvern Zeta Sizer because they've proven to be quite reliable and they're used worldwide by most leading laboratories, and that's something that we use as well as a characterization technique as well. As well as DLS or Malvern master sizer, I would suggest that you would also do some image analysis of your particles such as TEM and SEM, and for comparison morphological purposes as well. Where can we get or purchase lipid libraries? The most comprehensive lipid library. There are companies available who have these available. I think you just need to do a little bit of research into that to find out. Or you can develop your own as well, and that's the point of a system like this, is enable you to have that flexibility to create your own library and test out what's best for you. Because even though these companies may be able to offer you a library, doesn't mean it will necessarily suit your final application. So you would still need to do several tests downstream to identify whether it's the right one for your API. So you're asking 20 types of each lipid. Is it practical to purchase, for example, 20 to 30 lipids for each lipid type? You mentioned total of 4 lipid. So can we purchase, for example, I mean, you certainly can purchase as many lipids as you like, but the key idea is to investigate what each type of lipid is going to do to your formulation. What is it that you at the end would like? This is where you would need to narrow it down. I think not necessarily having lots of lipids is the key answer. It's refining one formulation with different types of lipids. So this is helper lipids, pegylated lipids, cationic and ionizable lipids. It's finding the right combination would be key, and also the concentration in which you use each of these and which solvent you disperse them in is also an important consideration. So I would do a little bit of research around that, narrow it down to 3 or 4 candidates for each, and carry out a design of experiments where you can easily detect very early on where your key trends lie. So what are the channel dimensions used? So different chips have different dimensions. For the app note that will be coming out, I believe we've used 100 micron and 190 micron because we've used a dilution step in that work. So the diameter of the junction between the aqueous and the organic phase is 100 microns for the first chip and 190 for the second chip. But we have different dimensions available. So again, this is adding more flexibility for the user as well. You can use different channel sizes. We can go up to 275 microns, for example, or even as low as 50, 30 microns. But it's just trying to work out the throughput, balancing that with what's right for that particular formulation. So there's another one. What is the maximum encapsulation efficiency that can be achieved? So again, we've had various customers who've already got our systems report back to us with 99% encapsulation efficiency, and this is particularly to do with RNA as well as other APIs as well. So it can go as high as 99%, and it's just a matter of trying to optimize your parameters as well as your formulation to achieve that. I think we are out of time, so I think if there are a couple of questions still remaining, I believe, and we will get back to you with the questions that have remained unanswered. I'll try my best to get back to you with answers on those. But thank you very much for attending. The webinar will be available for you to view on our website if you would allow just a couple of days for it to become available, and you should be able to refer back to it at your leisure. So again, we have some handouts. If you haven't already downloaded, please feel free to do so. And if you have any feedback and suggestions and any further questions, I do recommend that you get in touch with us, and we would be happy to help. So once again, thank you very much for attending today, and I look forward to hearing from you soon. Thank you.